So today we're going to be learning how to draw DNA replication. So the first thing we're going to do is we're going to draw a DNA strand. And this DNA strand is a two-dimensional strand that's already been cut up open. Usually DNA strands that are two-dimensional look like a ladder that's just laying flat. So this triangle that is right in the middle or towards the center is called helicase. It's a type of enzyme which does the cutting up of the DNA strand. We also are going to draw dots around the entire DNA, and these dots are single-stranded binding proteins, also known as SSBP. So the purpose or the main use for helicase is to break the H bonds within the DNA, and the SSBP are what keep the replication fork open. So this whole entire thing you see in the middle is known as the replication fork, where the replication happens. So now we're going to label these two circles that are on the side. So these circles have a funny name. The T is topoisomerase, and the G is gyrase, and they both relieve stress. They're not as important as helicase, but they are essential as well. So now we're just going to draw out our nucleotides on the side. So they're supposed to be based on the 3' and 5', which will be written later, but the bases are always within the DNA themselves. And we're just putting the phosphate groups on the sides. Now we're going to label 3' on the right and 5' on the left. This is known as phase 1, and now we're going to be moving on to phase 2. So for phase 2, we're going to be needing primase, which is what creates a bonding site by dropping off RNA primase. So we're using a PR within a square to abbreviate that in the pink color, as you can see. And I like to think of it as a landing zone for the next thing that's coming up, which is known as DNA polymerase 3. So DNA polymerase is what writes a new sequence for us. It's what changes 3' and 5', and it changes it to 5', 3'. So here we are adding the DNA polymerase 3, and it's going to continue the rest of the strand. So the DNA polymerase is the blue. As you can see, it reads 3', 5', and it writes 5', 3'. A way of remembering this is thinking you're reading 35 pages and writing 53 pages worth of commentary, if you're in IB English. So this is what's known as phase 2. Now we're moving on to phase 3, and I forgot to write that, but this is phase 3. And the next thing we're going to have is DNA polymerase, which is what checks for any errors, and it substitutes RNA for DNA. So basically it's going to change our pink that we have, which was the primase, or also known as RNA, and it's going to change it into the DNA polymerase that we need. As you can see, we don't need RNA, we need DNA. So that's what that worked for. And now there's another error, which is this gap that we have in between the strand. And we need someone to rescue us, so that's what this little hospital sign is for. This little cross with the L inside is known as ligase, and this is what connects the phosphate backbone together. As you can see, this is the ligase acting, and ta-da! We have a complete strand. And this is what it looks like when it's all done on the bottom. So now we have to do the top strand, and this is a strand that goes in the opposite direction. As you can see, I already labeled 5' and 3' because it's the opposite direction. So of course, like we did before, primase came in and it dropped off its RNA primase, and DNA polymerase 3 came in and also dropped off its DNA. And it's, as you can see, the arrow is pointing to the left. It continues on to the left. So because this is different, there is a word for it, and it's called Okazaki fragments. I'm so sorry. It is called Okazaki fragments. So these fragments are put together by ligase as well. So once again, ligase to the rescue. That is all. Thank you so much for watching.